Welcome to BioLite event. This is Saikumar. I am one of the Bioclues member. I worked as a research associate in DuPont Knowledge Center, Hyderabad. I worked in molecular biology lab, and my job responsibilities include high throughput cloning, transformation, and development of transgenic plants, which includes genotyping and phenotyping experiments. Before joining to DuPont, I worked as a research fellow in Directorate of File Seed Research. There I worked on reduction of endosperm's toxic proteins, resin and RCA in castor using artificial microRNA technology. As we all know, castor is one of the important oilseed crops, and the economic part of the castor is oil. This oil is used as a jet fuel as well as engine oil in many vehicles because of its high velocity. And the castor cake, after extracting the oil, the cake can be used as animal feed because of high nutrition content, but the presence of these two toxic proteins limiting the utility of this castor cake. So taking this as an objective, we plan reducing the toxic content in the protein in the plant using artificial microRNA technology. This AMRNA technology is the most advanced technology compared to other gene silencing technologies, and these resin family protein genes are around 28 copies present in the castor. So for designing the artificial microRNAs, I had a chance to learn and use different bioinformatics tools, and at the same time I had a chance to participate in the Bioclues bioinformatics training program, which is organized in the Hyderabad. And there I got a chance to learn different tools, and in the training program I got an opportunity to learn. I got a lot of confidence in using different tools. And apart from that, I worked on in silico identification and characterization of the plant promoters. I used different tools like plant care, plant promoter, place, like different tools to identify cis elements present in the promoters. As we all know, the presence and position of cis elements are very important for the promoter activity. And apart from this, I worked on codon optimization and identification of microRNAs from the different sequence databases. These microRNAs are smaller RNA molecules which are involved in the negative regulation of the genes and which are involved in the different developmental pathways in the plants as well as the animals. And I used different bioinformatics tools to identify these microRNAs from the sequence databases like EST sequences, GAC sequence. I used whole genome sequence also to identify microRNAs from the plants. And with the tools, I could able to successfully identify microRNAs from the sunflower, safflower, castor, and pigeon pea. With the help of Bioclues, last year I could able to successfully publish pigeon pea microRNA paper in Australian Journal of Crop Sciences with the help of Prashant. And this year, recently I submitted neem microRNA paper to the Gene Journal, which is in the communication stage. And apart from this, recently I started working on SSRs, simple sequence repeats associated with the microRNAs. And for this project I am working with the Anuj. He is also Bioclues active member along with the Prashant. For this purpose, we have downloaded all precursor microRNA sequence of Arabidopsis and subjected them to different bioinformatics tools for identification of SSRs. Initially we were working on Arabidopsis. Later we would like to translate this work in other crop plants. And apart from this, recently only I started working on miRNA PEEPs, means miRNA encoding peptides. And till now, microRNAs are coming from the non-coding part, and they don't code for any proteins. But recently, scientists have proven that microRNAs also code for small peptides, which are known to increase the expression of microRNAs. So recently, we started working on miRNA PEEPs from the Arabidopsis. For this purpose, we have downloaded all pre-miRNA sequences and surrounding regions, and we are subjecting them to different bioinformatics tools for identification of miRNA PEEPs. And these two works I am working along with the Prashant and Anuj. And with this one, I would like to conclude my work. If you have any doubts in my work, please contact me. My email ID is saikumar@bioclues.org. And I would like to congratulate all participants in the Bioclues event. And I would like to thank Prashant, founder of Bioclues, and other organizers for giving this excellent opportunity. Thank you. Bye.